Anyway, I want to talk to you about some of the research that my lab has been pursuing night and day for the last, I guess, about 2 months now. And this is a collaboration between my lab and then the lab of Reg Penner and Phil Feldner you're going to hear about as this goes on. By the way, don't hesitate to interrupt me if you have any questions. Sometimes it's easier just to get the question out of the way right there. So I also need to start with the important disclosure. Reg and I started a company called Phage Tech, which licensed the technology I'll be talking about, and they are developing products related to this research. And we have an approved protocol with the Conflict of Interest Oversight Committee at UCI. So that includes, for example, the data before it gets published is vetted by my colleagues before we publish it, just to make sure, for example, that we're not hyping the data for commercial purpose or something like that. I wouldn't do that anyway, but that's the way this works, and I think all of us are familiar with that. Okay. This is the project that I proposed to the COVID craft team some time ago. In short, what we want to do is map out how are antibodies recognizing this SARS-CoV-2 virus over here on the left, which I guess all of us are now familiar with what it looks like because it appears almost daily in national newspapers. We're doing this in 2 ways. One way is we're applying phage display, and I'll tell you more about that in a moment. And the other way is with Phil Feldner's lab, we're using protein microarrays. And then we take the winners from this, the antibodies that we learned to recognize from this process, and bring them over here and wire them into electronic circuits. And we're using this to develop a very rapid antibody-based test that we originally developed for cancer detection and cancer biomarker quantitation, but now we're going to apply it to antibodies. And I'll tell you more about that in a moment. Okay, so the goal really is to start looking at different stages of the virus and then hopefully quantitate exactly how much antibody someone has to see, are they going to be capable of neutralizing virus or not? That's the ultimate goal. Okay, here are the immediate aims, and here's a kind of an early version of what our sensors look like. But the goal is to figure out where do these neutralizing antibodies best attach to the virus? How is the immune system responding in a way that we can then use for a diagnostic? So in other words, I need to know not just where are they attaching on the virus, but also are they attaching to some spot that's different to the spot that MERS or SARS-CoV-1 we're attaching to. Those are 2 related coronaviruses. And we need antibodies that are going to distinguish between attachment in one spot versus another. And then we want to turn that type of antibody recognition into an entirely new type of diagnostic, one that's going to be fast, and I'll show you 60-second measurements in a second, and capable of quantifying antibody concentrations. Currently, this is not being done, but being able to quantify this antibody, not this antibody, that antibody, those are currently not available. And again, identifying specific antibodies. By putting all this together, we're expecting to have a much more powerful serotype diagnostic or antibody-based diagnostic, because it will give us information that currently is not available, and it'll give it to us faster, which should allow us to deploy it more widely. So the third part of our investigation is testing this with samples from UCI ICTS and then other samples from the Feldner laboratory. Okay, more immediately, the information we're generating is going to be novel, and we would need to get this out here, out into the world. So at every step, we're thinking about places to publish. If we start epitope mapping antibodies, publish that. We've developed a new type of diagnostic. Reg is already starting to write a paper on that one. And then testing samples from patients. All that will lead to new knowledge creation, and that's ultimately what drives us as academic scientists, in addition to the imperative that we get something out to help the world about this. Okay, so let me show you some preliminary results that were the basis for this proposal. And this goes back to research that Reg and I have conducted, Reg Penner and I have conducted over the last 15 years here at UCI. And most recently, it was funded by the National Cancer Institute to detect biomarkers associated with cancer in urine samples. And here I'm showing you some data from that. So this is detecting a bladder cancer biomarker called DJ1, and we're detecting it in 60 seconds in a quantitative manner. These are different concentrations of the biomarker in patient urine, and it's highly specific. That's shown in these controls down here. It also has low error. Look at the error bars here. It also has very low coefficients of variation from device to device. We actually are hitting what's FDA required, even though we're building these on the benchtops at UCI. So they're not even being manufactured, but we're hitting the FDA's requirements for coefficient of variations. It's also generalizable, and that's the important part, because then we can generalize this to detecting antibodies. And then finally, this is the challenge for us. We've only detected small proteins with this. And I'll tell you today how we're starting to address moving away from small proteins. And I should find, before I move to the next slide, let me just note that we published these results in peer-reviewed journals, the best journals in our field. Okay, so it's not just starting a company and getting it out that way, but also publishing new knowledge that we generate. Okay, so what we did is we started with SARS-CoV-2 proteins. And remember, our goal is to be able to detect antibodies in a patient who has been infected with this virus. But I want to do a little bit more than that. I want to detect antibodies that are specific for CoV-2, not the first SARS, and not MERS, again, a closely related coronavirus. So we did some bioinformatics. We then divvied this thing up into our expectations of potentially good antibody epitopes that are color-coded on here. These are surface exposed. They're unique to this virus, not other viruses. And then we put them on the surface of a different kind of virus called a phage. That's a harmless virus. And we're doing that because then the phage acts as a carrier to lock them into our electronic device. Okay, so we did this with a bunch of different proteins. These are 3 different proteins from SARS-CoV-2. Here's a 4th protein. This one is particularly interesting. This is the spike protein. That's the protein that's sticking out on the surface of the virus that literally does look like a spike. It's the tip of the crown when you look at a cross-sectional view of this, or 2-dimensional view of this 3-dimensional virus. But again, we divvied this up into a bunch of short peptides that we're calling epitopes, and then we display them on the surface of the phage and then incorporate them into sensors. I should note here that this is a collaboration with the Feldner Lab who are also identifying different epitopes. And so the 2 of us working together can cover greater surfaces. We're focusing on short epitopes that have a greater chance of being specific. And then Phil's laboratory is very nicely screening the whole viral genome, the whole viral proteome. And we're going to hear more about that in a second. So I won't talk very much about that today. Okay, let me just point out this one epitope over here. This is in something called the receptor binding domain. And on the next slide, that's going to be important. So specifically, we've now been doing a bunch of ELISAs using patient serum and using commercial antibodies and mapping out where do those antibodies from serum and from commercial antibodies, where are they binding on these different proteins? And so, for example, here's one data point. So in this ELISA showing binding to a commercial antibody that's known to neutralize the virus, here it is binding here to that C21 that I showed on the previous slide. And that's really exciting to us because that's part of the receptor binding domain, a unique segment of SARS-CoV-2 that will allow us to have specific recognition. Okay, now the next challenge for us is going from detecting tiny little proteins to much bigger antibodies. And just to give you an idea of scale, the antibodies are something like not quite 10 times bigger, but close to 10 times bigger. And so that's a little bit of a challenge if you spend a lot of time building a device that recognizes small things. So good news is this has been solved in the Penner laboratory by these talented graduate students who are listed here. And this is some very early data that I'm showing you from our devices. This actually required a new type of device architecture for us. And I'm showing you some SEMs with our new device architecture. And this is what I'm showing you is the very, very surface of the electrode that's going to detect the antibodies. Oh, let me back up and just explain a little bit more. So what we're doing is in red, those are the phage that are incorporating our epitopes. These will then be wired down directly onto this electrode surface composed of this conducting polymer called PDOT. And then we send electricity through the conducting polymer and it goes through the viruses. And if antibodies bind in green, those little balls are meant to represent antibodies. If antibodies bind, that changes the conductance, the impedance going through here. And that's something we can read out very readily. It actually changes resistance as well. Okay, so this is exciting to us, but again, it required us to invent a new type of architecture on the surface, basically nanoengineering of this surface. And this is a 1 in 5,000 magnification. Okay, so what's next? What I've shown you is really preliminary results, but I think they're exciting preliminary results and they're publishable preliminary results. I've shown you that we're getting good at measuring antibody concentrations, and we next have to do this in as many SARS-CoV-2 samples as we can get our hands on. That's going to be a challenge. I think the audience, some of the audience members are aware of that challenge. Then we'd have to combine our best epitopes together with the VBRs and again, measure antibody concentrations in serum. There is the perennial need to raise more funding. That doesn't go away. We're still working on that. And then we're doing this in collaboration with our corporate partner, our industrial collaborators at Phage Tech. They're going to then take the designs that we make, license them from UCI, validate and compare our sensors versus commercial testing. We're leaving that to the company to do. Companies are much better at that sort of thing. In addition, stuff like engineering for manufacturing and then setting up a user interface, that's really a commercial goal, and it makes sense to collaborate with the company to do something like that. Of course, they're always in the mode of raising more funding as well. Okay, let me end by thanking people who made this work possible. On the left, I've listed all the members of my team, and I'm really blown away by how dedicated they are. They've been working in shifts, night and day, 6 days a week on this project since the beginning of this crisis. And we've operate on Slack and pass data back and forth. Every day I get to watch the data from home, and it's been amazing to see their work being generated. In addition, I'm really grateful for the collaborators that I've acknowledged throughout the talk. Finally, this wouldn't be possible without the award from the Craft COVID funding mechanism. Thank you very much to the reviewers and all of you who worked so hard on setting up that mechanism. I know that a lot of investment went into this, and the researchers at UCI are really grateful, and myself are really grateful. So here's a little picture of my team. And in happier days, we were funded by these organizations. Okay, let me stop here and take some questions.
Okay, thank you. So we have about 3 minutes for questions. I hope all those people are maintaining social distancing while they're working 24/7.
Yes, they are. Actually, they definitely are. It's we have a whole protocol in place that I've shared with other researchers in thinking about how to reopen our labs. But I can tell you in short that we're operating at about 250 square feet of usable space per researcher. Everyone's widely separated, everyone's wearing masks, et cetera.
Okay, sounds good. We're going to hear more about that this afternoon and ramping up, I think. Okay, so open for questions. We followed a pretty informal format, so please just come forward with questions.
So this is Eric Berman. Greg, that was really good, really interesting approach and wonderful technology, as best I understood it. I'm wondering, you know, it's the same sort of thing as Phil has been concerned about with identifying some of these optimal antibodies that will also identify specific proteins or parts of the proteins. Is there anything you would see that would move towards a possible vaccine based on this?
I think you're absolutely right, that identifying the perfect epitopes then would be really cool to develop a vaccine for, or better yet, an antibody. So there's another segment of my laboratory, I didn't tell you about this today, but I have a whole separate proposal where we're going to do phage display with antibodies displayed and then select for binding to those epitopes, because then that would be a therapeutic for the virus. And I'm equally excited about that. Thanks for suggesting it, Eric. It's a great suggestion.
Hey, Greg.
Hey.
That was really great. I had several interrelated questions. One, have you been able to, do you envision being able to look at correlate the antibody responses that you're measuring with any T-cell responses in these patients? Because I'm curious, I'm sure you saw the Shane Crotty article that came out in Cell looking at T-cell responses and antibody responses and the cross-reactivity with other SARS or coronaviruses. So that's my first question. Secondly, I assume you can measure the isotype of the antibody? And then,
yes.
Okay, and then thirdly, well, I'll let you answer the first question, then I'll go to the third.
Okay. Great question. Tom's obviously a very sophisticated audience member. So he knows that to have a really effective antiviral response, you need both T cell and antibody. And unfortunately, Tom, at this moment, we're just totally focused on antibody response. I would love to be able to do T cell, and I should talk to you about that. We're going to need to bring in a collaborator to help with that.
Okay, great. And then my last question, you're looking at the potential cross-reactivity with the MERS-CoV and SARS-CoV-1. Have you also considered doing some of the more common upper respiratory coronaviruses like 229E, et cetera, because that might also be very important?
No, it's a great suggestion. On our, we've done it at a bioinformatics level, but not thought about it, how to actually make that work on a testing level. I'd like to talk to you more about that.
Yeah, sure. That's great work. Congratulations.
Thank you. Thanks a lot.
Greg, that last point's a really important one because, you know, as you look at the diagnosis, you need to be able to distinguish between a past coronavirus infection, you know, somebody who may have in November had 229E or OC43, because those are basically false positives, which can lead someone to expose themselves, or alternatively, if it were done in a detection method, you know, you'd be worried about someone being called positive when they're not. So there is a problem there.
Okay.
And I don't know, how would you incorporate multiple? Could you do it on one chip, or would you require multiple chips?
Yeah, so we're not there yet, but our goal is to eventually have a chip that has multiple recognition sites.
Yeah,
and
sort of I think we can do that. 96-well plate chip.
Exactly.
Yeah.
Yeah, and actually we're gearing up to do exactly that. I have part of my team is actually 3D printing these electrodes and working towards that type of thing.
Okay.
So it's actually really cool. It turns out you can 3D print electrically conductive materials now, which is really neat. It's a whole thing I'm learning about. Okay, anyway, thanks for the good suggestion. I've taken note, and we'll do our best to try to incorporate that. But I think it starts to hint at why current antibody testing is so inadequate. And it starts to suggest really where the field needs to go to be more successful at responding to this pandemic.
Absolutely. And at the danger of exposing a weakness in the system, could you maybe comment on where you're getting your biospecimens?
Yeah, they're coming from Phil Feldner's laboratory. We're really lucky to collaborate with him on this. Okay. I would love to receive some from ICTS. We've applied to the biobank 2 times now, and I'm hopeful that the doors will be opening there.
I think that's ramping up as well as other things. So so we look forward to hearing more about the practicalities of that.
Awesome.
But thanks very much. That was great.
You're welcome. Thank you.